Hello students, I am Dr. Shilpa Zadhar, Head Department of Microbiology, Sathe College, and I am basically a microbiology teacher teaching paper 3, Microbial Biochemistry, Unit 2. I am going to teach you today bioenergetics. Every single cell for survival requires energy, and as a result of which it needs to perform certain kind of chemical reactions which will help them to make energy which is in the utilizable form. There are microbes which can be differentiated depending upon the kind of reactions they bring about to make energy. The reactions generally belong to a category called as phosphorylation, where a phosphate rich compound is broken and it is basically given to a compound which is complex and is undergoing some kind of a catabolic reaction, as a result of which that particular compound also gets phosphorylated. Over the process of catabolism, it gets broken down to simple materials, as a result of which energy is made and ATP is produced. So, let us see the type of reactions a particular type of organism can bring about. The first is cellular respiration, which is also known as substrate level phosphorylation, which will basically make use of energy which is in the chemical form like glucose, which is essentially complex, not as simple as carbohydrate, and glucose when will get broken down in the presence of ATP, it will by itself also generate some particular amount of ATP. For example, glucose oxidation basically yields around 2 ATPs, and glucose fermentation via EMP can yield 7 ATPs. The next is aerobic cellular respiration, which is being brought about by organisms which are strictly or obligately aerobic in nature, and that is why they will require oxygen. Whenever an oxygen molecule is available into the atmosphere, organisms are generally known to carry out electron transport chain which is oxidant dependent, and that is why this kind of reaction is called as oxidative phosphorylation. The transfer of electrons from one carriers to the other develops a proton gradient, and with the help of this developed proton motive force, a molecule of ATP can be synthesized. This way you can yield energy. The next is photosynthesis, which is being carried out by only a specific group of organisms which are ideally photosynthetic in nature, and these organisms contain the pigments which are required for photosynthesis. Pigments are either present into the phycobilisomes, or certain types of organisms also have some areas called as thylakoids, and as a result of which you will see the photons will be captured by the organisms, basically the pigments, and again there will be an electron transport chain that will happen, as a result of which by using the light energy, carbon dioxide and water, these photosynthetic organisms are capable of synthesizing their own carbohydrate requirements. That is why they are also called as autotrophic in nature. So, here is an example of a substrate level phosphorylation where the ATP is formed when an enzyme transfers a phosphate group from a substrate to ADP. It takes place in cytoplasm and mitochondrial matrix as well. For example, the TCA cycle. Direct phosphorylation occurs in Krebs and glycolysis cycle. Basically, glycolysis is known to yield 7 ATPs and TCA is known to yield somewhere around 28 ATPs. Oxidative phosphorylation, it is a metabolic pathway in which cells use enzymes to oxidize nutrients, thereby releasing energy which is used to produce ATP. It takes place inside the mitochondria. Indirect phosphorylation occurs in the electron transport chain, and somewhere around 34 ATPs are formed. You can see there are 4 complexes which are present here, and each of the complex is capable of pumping one proton for each electron, and generally the electron transport cycle takes place with the help of 2 electrons. So, 2 electrons, 2 hydrogens are also pumped, as a result of which there is a hydrogen pool that gets generated outside the membrane, and this leads to the generation of proton motive force. So, the hydrogens are recycled again back into the cytoplasmic membrane, where there is presence of an enzyme called as ATPase, which will basically couple the entry of hydrogen to the ATP synthesis. Next is photophosphorylation. In this process of photosynthesis, the phosphorylation of ATP to form ATP using energy of sunlight is called photophosphorylation. Occurs in the stroma lamella. Basically, the pigments for quenching the light in the form of photons are present here. Stroma and lamella are ideally present into the plant cells, whereas in the photosynthetic prokaryotes they are ideally the chlorophyll pigments which are present into the membranes. Cyclic and non-cyclic are the two types of phosphorylation. Yes, you can see both the types. Cyclic is the one stage process and non-cyclic is a two stage process. So, you can see on my right hand side there is a non-cyclic process, and similarly like an electron transport chain there are also the electron carriers which are belonging to the group of phycobilins, and then there is something called as cytochromes and plastoquinones. So, similarly like that, and the electron transport chain is happening. At the left side you can see a photophosphorylation which is cyclic in nature. So, continuously whatever electrons are liberated at the end of this particular type of a cycle, that same electron can enter back into the photosystem, and that is why it is more cyclic in nature. You can see the last electron acceptor is not oxygen here, alright. It is generally a kind of a metallic molecule, and as a result of which it is different from the regular electron transport chain that happens into the aerobic organisms or oxidative organisms. I hope you have understood at least the first 3 levels of phosphorylation mechanisms. Practice it, and I will see you tomorrow. Thank you.